Good morning, everyone. Hope isolation is going well for everyone, our social distancing. Um, I'm here at a very, very quiet museum today. There is literally no one else in the building right now. So I'm down back in collections. I'm in the collections where we have our vertebrate collections where we spend a lot of our time. But today we're going to do something different. We're coming up to a place where you see a whole bunch of boxes. It's a whole bunch of unopened boxes, and then there's a whole bunch of J.C. Penney's clothing drawers that were given to the museum sometime many years ago. And this is our area where we have a whole bunch of fossil plants. The university, for a number of different times in its history, has had somebody interested in paleobotany. A lot of the plant taxonomists or botanists in the biology department have also had interests in fossil plants, and so there's been a lot of collection of fossil plants in the past, and we have all kinds of really a pretty large collection. There hasn't been a lot done with the fossil plants as far as being getting them digitized, identified, all that sort of stuff, but we have a pretty large collection. So you can see there's all kinds of stuff to do. If you open up a couple of drawers, which I decided to do, you can see that we have a bunch of rocks, and if you get down and look really closely, goodness gracious, you see all of the darker markings. That's all plant material that's been preserved in clay, which is cool, and we get all sorts of different things. For example, over here, you can start to see stemmy, woody pieces, and you can also start to see leaves, like right along here. So a lot of times, leaves are often impressions, but we also get sometimes when there's a little bit of the original plant material that is still fossilized. All of this stuff is Cretaceous, and as we get to some bigger pieces, you can start to see whole leaves. And let's see, oh, there's a good one here. We've got some that are a little bit, have all kinds of interesting different morphology. And as we were talking yesterday, I was showing you a bunch of leaves that I had collected from when I was 13 in Iowa, and almost all of the leaves had some sort of denticles, serration, tooths in their margins. And just as we're in our first drawer of looking at some fossil leaves from the Cretaceous. These are all Cretaceous age, around 100 million years old or so. You can see that they're pretty much entire margined, right? Very cool. So you don't see a whole lot of denticles or teeth on serrations on the edges of these particular leaves. Already gives us a hint that there's something different about the Cretaceous than there is about leaves that are around today at around this latitude. Here's another really cool drawer randomly opened, and you can see a whole variety of different sorts of plant material. As we get really close, there are some different sorts of leaves, and there's quite a huge variety of things that are coming from trees versus things that are coming probably herbaceous sorts of things that were growing up from the ground that were not woody or not tree-ish. So we have cabinets and cabinets and cabinets of lots of fossil plant material. A lot of these drawers have a lot of the samples that, man, we've been talking about the plants in the herbarium, lots of things that are collected ending up in newspapers. So we have lots of, it's the same thing with fossil plants. Hey, we even have something from Hayes Daily talking about plans for pick and haul. We even got a little bit oh, these are actually the university newspaper, back when the university had a newspaper. Let's see, where were these specimens collected in Kansas? We've got them from a number of different places. Hoisington area is one of the places that we've had a lot of these things collected from. We've had them collected from Wilson Lake area. We've got them collected from a whole bunch of different areas. As I'll maybe cover a little bit more as we get to it. One of the things that's interesting too is, you notice we have a whole bunch of boxes on this side that are labeled petrified wood, 80 specimens. That's kind of interesting. I open up one of these boxes to give you an idea. And yes, we have lots and lots of just small chunks of petrified wood. There's not a lot sort of unique or special. One of the interesting things about fossil woods is you have to you can slice them open, and there's actually a great website that's associated with the North Carolina State where they have a whole catalog of fossil woods. If you slice open your fossil wood, come on now, you can actually measure and look at the growth lines, growth rings in the wood to actually identify things. So it is actually kind of a cool and possible thing to do that. We have some larger, cooler pieces of fossil wood where we start to get whole logs. This is kind of cool because this is a log about 2.5. Let's try and stay connected here. All right, 2.5 feet in length. And you can really start to identify and see the lines and the structure in the wood. You can even see some things like some knots in the wood. We might have had branches or things growing out in different directions. And this is a very silicified piece of wood, but it's kept a lot of the texture of the wood. And let's see, oh, there's even more evidence of knots where you've got things, branches, things growing out vertically away from the main tree trunk. So this is a very large, very cool piece of a tree. Still very silicified. If it's petrified wood, but sometimes you get fossil woods that are still have a lot of lignin or the organic material of the tree, as in this one. And so this is still very soft and woody. You still see a lot of the organic carbon in it, and you still see some of the texture of the fossil wood. This is kind of stuff, it's like coal. Coal is made out of fossilized wood that gets heated up and burned, right? So it's leftover lignin of fossil trees. And so when you're out finding coal, you're finding stuff that's basically similar to almost to this. This has got a little bit of silicification, but it still has preserved enough organic material in the tree. Whereas if we come over here, we talked about a few days ago that sometimes in the Cretaceous Seaway, you get wood that is float. So it's driftwood floating through the ocean. And when that happens, as you get really close, you can see the texture of and the grain of the wood. But you see all of these little in the wood. Those are actually borings or trace fossils of shipworms called Pteridolites. So it's riddle in here are basically little borings of things that would drill into the wood, little clams, shipworms, and that's the little holes they left as they were making a place for themselves to live in driftwood in the Cretaceous Seaway. So, but when you get, if the internet stays connected, you can see really cool the grain of the wood in this piece. We have all sorts of other sorts of cool bits of fossilized wood. A lot of it's even, we have a typical piece that you find in Arizona in the petrified forest, which is highly silicified, and you get all the really cool, fun colors. In petrified forest woods, you oftentimes don't see the grain of the wood on the inside of the pieces, but if you go to the outside, you actually can see sort of texture on the outside of the logs, which is kind of cool. So you can tell it's a tree, even though when you come up to the top, it's really beautiful, as much of the cool texture of the wood. Whereas some things are so well preserved, boy, you can see the outer growth of the tree. You can tell sort of what kind of a fun tree this is, and you also can see. We're here a little bit, looking at all these more boxes and boxes of things, and we have also, here's another really cool tree trunk, and you can tell that this is, flip it around. Gives you a little chance to see how big this is. Use myself as a scale a little bit. I can only go so far back. Doop, 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 there we go. But this is kind of a fun tree. And again, you can see large knots where you had different branches, and you can actually see down into the tree. Maybe you had something living in here at one point in time. So they're pretty spectacular tree. See, I could imagine finding a little vertebrate that died in here. And that has happened sometimes. Obviously, that's pretty rare. Didn't happen in this case. At least I don't see any bones down in there very easily. But what's cool is the history of plants a lot of times is in fossil woods. We have actually like fossil forests that are preserved in different places, petrified forest being one in Arizona. We have high latitude ones found up in the Arctic of giant forests, which is really cool. What kind of tree is this cool trunk from? That's a good question. I see Joe Thomason is watching. If you have an idea of what kind of tree this is, you can type it in for us, because I'm not exactly sure. So let's move along, looking at all of our fun things, and we're going to move over and start looking at some other fossil plants. So here we have some cool drawers of leaves that are Cretaceous, and these actually from Kansas, and not too far, not for more than 40 minutes or so from Hayes. And here are really cool because we have concretions or nodules, fossil leaves in them, and you can look really closely and you can see perfect leaves, and you've got all kinds of with really nice veination. You can see, internet, don't fail me now, veinations in the leaves, and they're all in these nice cool concretions, right? So, and what is that some of these concretions, you can tell, interestingly enough, have the shape of sort of a leaf, but you don't see the leaf yet. You can see the whole concretion. Now if we were to take this one and split it, hammer it and split it, guarantee you that you would find a leaf inside. All these concretions come from one single layer or level, so they were basically all fossilized at the exact same time. So this is almost like its own little reconstruction of a forest. As a matter of fact, here is a very large leaf. Put my hand in here for size. Give you a little bit of a scale. You can see the veination in the leaf. You have an entire margin on this, right? So it doesn't look like there's any serration. So this came from a rainforest, probably pretty warm. So the Cretaceous is accepted to be a lot warmer than we have today. We talked about looking at toothed versus entire margins, and that the warmer you are, the less serrations and toothing that you have. You have more trees with, and you can kind of see that in a lot of these particular leaves. There's lots of different, some of them get long, and you sort of, some of these I think even have drip tips on them. All right, but we have a whole bunch of these things. How do these concretions form? I'm sorry, when we get down in collections, sometimes our internet doesn't stay connected so well. A lot of times in, sandstones, when leaves get buried, the organic material, as water goes through, starts to initiate the precipitation of cements. And cements can be dissolved silica, sometimes it's dissolved carbonates, in this case it's silica, and it's the organics that start to initiate precipitation of these cements in between all of the grains of sand. So you start the growth of the precipitation around of the cement right around the leaf, and then it works its way away from the organics, which sort of cements the sandstone around the leaf, which then, as you get far enough away, the cementation sort of stops, and you end up with a concretion that's gone around all different directions around each individual leaf. And so that sort of gives you a nice little tomb for individual leaves within a layer of sandstone. And that's sort of what we have here, are all of the individual tombs for each leaf within a layer of sandstone, which is kind of cool. We can open up drawers, and we start to see a whole variety of different leaves and leaf shapes. And sometimes you get some with even some denticles on the margins. So they're not all the same leaf. We could identify a whole individual forest and sort of get an idea of its makeup. You know, I showed just with my collection, you can have 30 or 40 different trees and a little around the town of Osage. Well, this is one layer, so one little bit of a forest, and you can start finding, just in these few drawers, at least 15 or 20 different types of leaves, which is pretty cool. And sometimes you get them some more iron staining, so you could get the leaves that show up bright orange in the concretions. A lot of times they're the darker browns and reds, and you again can see the veination in the leaves, which is kind of cool. Right. So we have a whole variety, some project to figure out what is the actual composition of this particular forest based on all of these leaves from these concretions. And this whole locality was actually found by Dr. Thomason's wife, Nadine Thomason, which is pretty awesome. So it's... We also have some other localities that come from a ranch, and the mudstones instead of sandstones, and you can start to get stems and branches. Get my finger in there. So you get all the different. In this particular, you get a lot more of a mishmash of stems and different woody branches and things, as well as bits of leaves that are nice and shiny. So one of the things that's interesting, all of these plants come from, yes, Dakota sandstone, and that means they're at about 100 million years old. Show you something that's kind of cool. This is a picture from a publication that shows the oldest flowering plants on the planet. And this particular plant is called Montsechia, and it's 130 million years old. It's from Spain, and it is a water plant. So the earliest flowering plant, this has some seeds in it. So the oldest flowering plant is 130 million years old. Comes from Spain and parts of Europe. Well, we're in, and you don't really see a flower very much in it. But I said we were looking at the Dakota sandstone. And in this layer, with all this mishmash of stuff, we're talking about 100 to 110 million year old plants from here in Kansas. And what's this thing right here? Oops, can't get too close. That, my dear friends, is a flower. This is one, the first one Dr. Thomason showed me, but this is an actual flower. It's not just a leaf, it's not just, and this is another one that I found. These were the oldest flowers known on the planet 40 years ago. It wasn't till just a few years ago that they found older flowers in Spain. So what we have here are, in Kansas, in the Sternberg, are some of the oldest flowers on the planet, and definitely the oldest ones, I believe, in North America. So this is a pretty significant, cool collection. We find a few more. We could have a bouquet. So really cool plants here in Kansas, and really cool fossil plants. Some of the things we talked about is we could start to, yesterday we talked about figuring out mean annual temperature from leaf margin analysis. Another thing really cool about, especially some of these fossils, is some of these bits of leaves actually have their cuticle. Any way to determine colors of fossil flowers? In the past, I would have said no, but sometimes we can find, because there's organics, and sometimes organics, there are proteins that are coded to color, so I think that is perhaps entirely possible. We know we found colors from feathers of fossil dinosaurs. So yeah, I think that might be possible at some point. What of cool that I was wanting to point out here is, as Dr. Thomas had put here, extremely fragile cuticle exfoliating. So there's actual leaf cuticle from 100 to 110 million year old leaves. This is really awesome and kind of important for a couple of reasons. And it's another thing that we can do besides leaf margin analysis to understand climate in the past. Because cuticle will preserve these things. What do I have here? Close-up picture of a leaf that shows their stomata. That's all of these cool little cells right here. And stomata are the cells in a plant that open up and close to basically do two things. Let in CO2, because plants survive on CO2 via photosynthesis to create the carbon so that they can grow. So these things open up during the daytime to let CO2 in, so that the trees can photosynthesize and grow. But they also then, when they're open, they let water out. So they transpire water. So it's interesting because as the stomata are open, their water is evaporating or transpiring out of the leaves, which sucks water up from the roots and keeps water pumping up through the tree from the roots to the leaves because of the transpiration of water outside or leaving the stomata when they're open. Generally at night, the stomata all close up so that the tree won't lose water so fast. Why is this interesting? There's a couple of reasons. Generally speaking, if you're in environments where it's really, really dry all the time, plants have fewer stomata because they don't want to lose too much water. And where there's lots of more water available, a lot of times you have more stomata. Stomata also correlate to the amount of carbon dioxide in the air. And this is where it becomes interesting through time. A lot of times, if you have fossil leaves that come from a time period where there was more CO2 in the atmosphere than there is today, then those leaves will have fewer stomata in them because they can get the amount of CO2 they need with fewer stomata. Because plants are going to photosynthesize the CO2, and if you have a sunny climate with lots of CO2, they could end up almost having issues with too much CO2. So you have fewer stomata sometimes in time periods when there's higher amounts of CO2 in the atmosphere. That's where, getting back to leaves that have cuticle on them, and studied the number of stomata in fossil leaves, which is kind of cool. We add this to looking at the wood and the growth of the wood, and we can really understand a lot about the atmosphere and the climate. Just for a fun little kind of cool self-promotion, and let Dr. Thomason know that I had some work with plants in the past. I had a graduate student once who studied ginkgo. Ginkgos are cool because they've been around on the planet since 220 million years ago, back in the Triassic, and they're still around today. And I had a graduate student once take little ginkgos, and this is kind of cool. She worked at Duke, and this is the hyperbaric chamber at Duke. What's a hyperbaric chamber? And what does it have to do with plants? Well, hyperbaric chambers are chambers where people can go in, you can close the door, and there's all kinds of controls, and you can control the atmospheric pressure in. What's that useful for? Well, initially, a lot of scuba divers and things could go down and dive. And, you're not supposed to dive more than 30 feet deep without taking a long amount of time to come back to the surface. And if you dive too far and come up, you get nitrogen bubbles in your blood, which is called the bends, and that can cause you some heart embolisms, and you can have get bubbles in your joints, and you can end up killing yourself. We see mosasaur bones sometimes have evidence in their bones from the bend, of having the bends, so we know they were deep divers. Well, hyperbaric chambers, you can stick a person who's got some issues with the gases in their blood and increase the pressure, the atmospheric pressure in the chamber. to keep those, for example, to get those nitrogen bubbles dissolved back into the blood so that the body can deal with them slowly, and then you can work your way back out. It's a whole field of medicine of hyperbaric. What did we do? We grew these cool little ginkgo leaves in the ginkgo plants in these hyperbaric chamber, and we changed the atmospheric pressure, and we changed the amount back to what we think Cretaceous atmospheric pressure was, and we changed the amount of CO2, percentage of CO2 in that atmosphere, and we watched how fast the ginkgos grew. With increasing CO2, they grew 50 to 70% faster than they do at normal atmospheric pressure and CO2 levels today. And if you increased the percentage of oxygen and CO2 at about 1.5 atmospheric pressures, holy smokes, these things grew extremely rapidly, like double the speed of what they do today. So that's really kind of cool. It shows a little bit of the fact that high CO2 equals faster growth, but if you have high CO2 without higher oxygen, it can actually sort of poison you a little bit and slow your growth down. But you add both of those and plants will grow extremely rapidly, which is kind of important when you think about all of the huge plant-eating dinosaurs that were around in the Cretaceous. But plant growth sort of controls the rest of life on the planet. So understanding how they use their stomata, how many they have, and use the plants and what their stomata were, basically reconstruct how fast plants were growing and how that helped sort of control the populations and what kind of food productivity there was for dinosaurs and other animals living on the planet during the Cretaceous. So having flowers and having from our Cretaceous plants here in Kansas, we have the opportunity to do really cool things to understand the world back during, we could say, mosasaur time. I like to use mosasaur time since we're in Kansas instead of dinosaur time. But anyway, so we have all kinds of cool plants here in the Sternberg, leaf forests, and some of the earliest flowers in the world. So just another fun thing for us to see, things that are cool to see here in the Sternberg Museum. Did different atmospheric conditions back in the Cretaceous have any other effects on the ecosystem? Yes, would have affected changes in how much CO2 would have been in the oceans, would have changed the amount of productivity in the oceans for the base of the food chain. And we have, chalks are made up of tons of tiny little microorganisms of calcareous nanoplankton. So more CO2 means more plants in the oceans as well as faster growing plants on land. So very, very cool things that those conditions would have had. If we also had higher oxygen and higher CO2, it's not completely proven, but then you've got the ability for vertebrate life to be able to have more oxygen to increase their metabolism. So yes, atmospheric changes have huge effects on life in the ocean and on land, which is really, really cool. All right, let's see. Anyway, I think that sort of gives us a real quick introduction to paleobotany at the Sternberg from yesterday, talking a little about the herbarium. So I hope you have had fun sort of looking at the plants of Kansas and the plants in the Sternberg Museum over the last couple days. I know Ian will be back this afternoon at 2 from his house, so he'll have the home dome from home, or as opposed to. All right, so anyway, from our dome to your home. Have a great morning, have a great day, and stop in to say hi to Ian, who I believe will be talking about elasmosaurs this afternoon. All right, thank you much all. Have a great day.